artificial gametes, are they ready for the clinic? So, everyone knows Brussels a lot in Belgium, but we live in Ghent, we work in Ghent, so it's a very nice city also to visit. I'm originally from Bruges, it's also a very nice city. Disclosure, well, we get some funding sponsoring for stem cell research, but this does not affect the content of my presentation. I just want to situate our research that we are doing in our team. So half of it is about the topic today, which is the stem cells, which we want to differentiate towards gametes, but half we also concentrating on some translational research, such as failed fertilization after hESC, prevention of mitochondrial diseases, and using next generation sequencing for PGD purposes. So, stem cells. This slide shows an overview of the topics I will tackle today. I added one learning objective which was not in the syllabus. So I wanted to situate a little bit more why we want to make artificial gametes first. Then I will make some, show some slides about what kind of stem cells do we need for this. I will give you an update of what is already possible in mouse versus human, animal versus human, but it's mostly mouse. Then the fourth topic is a little bit more stem cell related, so if you're sleepy and you want to take a quick nap at point 4, if you're not so into stem cells, you can take a quick nap, and then I will wake you up for the final shortcomings and pitfalls which still have to happen. Okay, well, actually artificial gametes, we don't like to use the term so much because it's a little bit plastic. Actually, we prefer to use the term stem cell-derived gametes. So why do we need them? The first objective is, of course, to overcome infertility, which I will come back to on the next slide. But next to infertility treatment, it's also a very nice tool to get to know much more insight into the process of human gametogenesis. Because, as you know, it's hard to get in vivo material in human about the early steps of germ cell formation. And also, yeah, problems that can arise can give us new clues for treatment for infertility. Also, as you know, there is a very big lack of oocytes and sperm for donor banks. Yeah, oocytes is a much higher problem than sperm, of course, so it could provide a solution for this. Also, yeah, if we can make a lot of gametes, we can use them for training purposes in the lab, to learn hESC, for example. But also for research purposes, yeah, we need a lot of oocytes and sperm to do research on. Also for toxicity testing, drug screening. And also, yeah, it's a little bit controversial topic, but yeah, now it's a lot in the news. This germline engineering editing. So if you have stem cells and you can make them to gametes, you can perform a lot of gene editing also on the stem cells. So, but the main rationale why we want to make stem cell-derived gametes is for infertility. So if there is an absence of functional gametes in some patients, for example in cancer treatment, young boys or young girls who have to undergo cancer treatment, well, we can freeze, of course, but if they are prebertal, it's not a possibility. Premature ovarian failure, if there is no sperm, non-obstructive azoospermia, and others like Sertoli cell-only syndrome. So, yeah, there are some indications which give rise to absence of gametes. Also, yeah, there is some talk going on that we could provide stem cell-derived gametes for same-sex couples. So, for the homos or lesbians people, also for singles, it might be a possibility to reproduce. So if you have a man, you could make from the stem cells also oocytes and mix them together. But these are, yeah, topics that are still far in the future, I would say. So what do we need to make stem cell-derived gametes? Well, this slide shows a basic overview of where the gametes come from. So Of course, it starts the life with a fertilized zygote, and then it differentiates into the blastocyst, and then you have the three germ layers and all the derivates from the three germ layers, the mesoderm, endoderm, ectoderm. And the gametes come from a special precursor cell. It's the primordial germ cells. Well, this is a special form of cells because it's the only cells who undergo meiosis. And the other cells don't undergo meiosis, so it's the only cell which are haploid finally, the oocyte or the sperm. I'm a little bit sweaty. I will take off my jacket. So in vivo, you know, the process of gametogenesis. So you first have your precursor cells, your primordial germ cells, and these primordial germ cells then, depending on external signals, they will undergo spermatogenesis, in which they will form the mature sperm, or they will form the mature oocytes, so depending on external signals. So it's actually two big steps. So first primordial germ cell formation, and then the spermatogenesis or the oogenesis. This is a nice review from a group of Amsterdam, which actually shows the different routes to make gametes from stem cells. So they describe actually 8 or 9 for men, 8, and for the woman they describe 9 possible routes to make gametes from stem cells. But actually, yeah, they are all a little bit similar, and the main point is that you can derive gametes from embryonic stem cells, which I will explain now more in detail. So there are 3 essential steps in the process of making in vitro gametes from pluripotent stem cells. So pluripotent means that they can form each type of the body. First, you have to reprogram differentiated cells. You have to isolate the pluripotent stem cells from these. And then finally, if you have the stem cells, you have to direct them towards the gametes. So the question is this all possible in vitro? We cannot start from all the stem cells we already have in the lab. So normally we isolate stem cells from a blastocyst, so from the inner cell mass. If you isolate it and you put it in culture, after 2 weeks you have all these stem cell colonies, your embryonic stem cell colonies. But of course every embryo is unique, so it doesn't match. The genetic content is not the same as the patient you want to have gametes from. So what is the solution to have patient-specific stem cells? We need to use the technique of cloning, so it's somatic cell nuclear transfer, or we need the iPS cells, so it's the induced pluripotent stem cells. This is a scheme which shows it schematically. So if say patient Rudy comes to us and he doesn't have any sperm. So what do we need to do? We need to take a somatic cell from Rudy, let's say a fibroblast, skin fibroblast. We need to put it into an oocyte in which we first removed the nucleus. So now the oocyte contains only the somatic cell from patient X, patient Rudy. We artificially activate the oocyte, we make an embryo, and on the blastocyst stage we have the ICM, the inner cell mass, and from this inner cell mass we can make embryonic stem cells. And then the most important step is that we differentiate these embryonic stem cells towards the cells that we want to need. In this case we want sperm for this patient. So it looks all very science fiction and maybe far-fetched, but this is already possible. It's possible in mouse, not in human yet, but this whole process is possible in mouse. So I will give you some state-of-the-art of these 3 essential steps in the process. So first, the reprogramming. Well, reprogramming differentiated cells, we also know reprogramming in vivo. So there are 2 stages during development that you have reprogramming. So it means that the DNA methylation is minimized to the lowest level. So it's the stage of the precursor cells, the primordial germ cells, and it's the stage during embryo development when you have a blastocyst. There is also a genetic erasure, epigenetic erasure. But in vitro reprogramming, we mean that we want to make somatic cells embryonic again. So we all know this sheep dolly, I think. Next Tuesday, it's 20 years ago exactly, on the 5th of July that she was born. So this was actually reproductive cloning. So instead of making stem cells, they transferred back this embryo and they made live offspring. So this is reproductive cloning. But yeah, what we want to do is therapeutic cloning. So but it's the same technique. You first have to clone an embryo. So we need to aspirate, take away the nucleus of the oocyte. This is with staining, but nowadays you can use this polarized microscopy to visualize the spindle. You take it out, and instead of a sperm cell, you inject a somatic cell. These are cumulus cells that are frequently used for somatic cell nuclear transfer. And then you artificially activate them, and you have cloned embryos. We also did it in the past, some research on this, but we couldn't get to the blastocyst stage. They all rested, our embryos. Yeah, this is just an overview of how you make stem cells. So from the inner cell mass, you isolate inner cell mass, and you get your stem cells in your dish. So who was successful in making cloned embryos? Well, we first had this Korean guy, Hwang. He claimed to have produced cloned embryos, and also he derived embryonic stem cells from these cloned embryos. But it was all fake, apparently. And he was even in jail for a moment, so this guy, yeah, was not so good. But yeah, after him, they succeeded for the first time in 2013, so quite recently actually, to make cloned embryos, cloned blastocysts, and derive embryonic stem cells in human. It was the group of Mitalipov in the USA. Well, their success was majorly due to the fact that they could use very good quality oocytes, really from young donors. They could recruit young donors lower than 25 years, because above 25 years efficiency was much lower to make cloned embryos. And also, yeah, they modified the protocol a little bit. And this is now reproduced by a handful of labs, have now succeeded in making cloned embryos and derived stem cells from them. And still the main crucial factor is that you have to use donor oocytes from very young patients to make it successful. The alternative for cloned embryos is the induced pluripotent stem cells. So you may have heard of this guy Yamanaka. He received the Nobel Prize recently. So it's actually, instead of making cloned embryos and derived stem cells, it's actually more direct. So you have somatic cells, you transfect them with some genes which are important for stem cells, and after 2 weeks you have this kind of induced pluripotent stem cells. So nowadays they are investigating a lot which stem cell is better, so if the iPS cells are better or if the embryonic stem cells from the cloned embryos are better. There is some clues that stem cells from the cloned embryos are a little bit better than the iPS cells, but this is still under investigation. So for the final step, the in vitro differentiation, the most difficult step. So the first steps work. Both in mouse and human, we can make embryonic stem cells from cloned embryos, or we can make iPS cells. Now, the third step, also an important step, of course, we have to differentiate the stem cells towards oocytes or sperm. This slide is just, I show this slide to show that it's complex. It's not a thing that happens with a finger zip. So you have different stages. You have your primordial germ cells, and then you have to differentiate them in different steps towards the mature sperm or oocytes. So we want to mimic this whole process in vivo. that happens in vivo, we want to mimic in vitro starting from embryonic stem cells. You have the stepwise cells intermediate cells that happen, so stem cells, primordial germ cells, oogonia or spermatogonia, and then spermatocytes, or the meiosis procedure. And all these steps go together with some specific genes. So if you differentiate them in your culture, you can use all these genes to check what you have in your culture. These papers are the two most important papers in mouse. So these are the two landmark papers that have succeeded in making oocytes or sperm from embryonic stem cells. So it was from the same group, from Saito and Hayashi. So they first were successful in making oocytes from embryonic stem cells. And if you have oocytes, because before already some groups claimed that they had oocytes or sperm, the most important thing is if you have oocytes that you prove that they are functional. So you have to fertilize them, they have to form an embryo, and ideally you put back the embryo and you prove that you can have offspring. And also if you have offspring, you prove that the offspring can also generate pups. So they have tested all this and it works. Also in sperm, so the guys, don't worry, they also succeeded in making sperm, and they also proved that this sperm is capable of fertilization, embryo development, and live offspring. So 2011-12 mouse, it works from embryonic stem cells. There is one limit still. They still needed an in vivo intermediate step. So once they had the primordial germ cells, they transplanted them in the ovaria or in the testis, and then they got this functional oocytes or sperm. So this is a little bit limiting because, we still need an in vivo niche to fully mature the primordial germ cells towards the functional sperm or oocytes in mouse. One of their keys for their success that they demonstrated was that they used embryonic stem cells in a naive state of pluripotency. I will try to explain this a little bit, but it's very stem cell specific, so excuse me for that, but it's important to understand that you have different stem cell types. So they started from these mouse embryonic stem cells, or iPS cells. They differentiated them a little bit towards epiblast-like cells, and then they could form these primordial germ cells, and then finally functional sperm or oocytes. So I will explain a little bit these different states of pluripotency. If you're not interested, a short nap, you can do it now. So it's a historical overview of embryonic stem cell derivation. So the first mouse embryonic stem cells were established in 1981 already by two groups. So actually they called these stem cells later the naive embryonic stem cells. And in 2007, they could derive a new form of pluripotent stem cell, which they derived from the post-implantation embryo. So the first embryonic stem cells were from the pre-implantation embryo, so from the blastocyst, from the inner cell mass. But in mouse, you can also harvest post-implantation embryos in a mouse, and they could also isolate pluripotent stem cells, and they called this the epiblast stem cells. And afterwards, they called this state of pluripotency primed. The first human embryonic stem cells were a little bit later, 1998, and we didn't know if they were naive and primed. These subjects of naive and primed are only recently discovered. So now we know that they are primed, but we didn't know before. So again, to show you, because this is important if you want to learn something about stem cells, I want you to learn that there are two different states. So you have your naive ones derived from the inner cell mass of the blastocyst stage, and you have your primed ones derived from a post-implantation stage. So the human ones, which are they? Are they naive or primed? So they are also derived from the ICM. We cannot get post-implantation blastocysts in the human, of course, so they are also derived from the ICM, similar to the mouse. But they like the same factors in the medium like the primed ones, like the mouse epiblast stem cells. Also they did a lot of gene expression analysis and other molecular analysis, and they actually showed that the human embryonic stem cells are primed. Primed means that they are already a little bit differentiated, so they have a propensity to already start the differentiation process. The naive ones are very homogeneous, they don't differentiate yet. Yeah, there are some advantages and disadvantages in these different states of pluripotency. So the naive ones, the advantages are here. One important thing is that they contribute very efficiently to chimera. So chimera is one of the most important proofs that you have pluripotent stem cells. They like single cell passaging, so you can easily culture them. They are much easier to handle in culture because you can do single cells, so you can make millions of cells very fast and easily, and they are less prone to differentiation compared to the primed stem cells. Yeah, it's a very chaotic slide, but just to show that the primed stem cells, the human ones, yeah, they have some propensity. So some of your embryonic stem cells will rather efficiently go to blood cells. The other ones will more like to go to neurons. So they already have some bias towards some cell type. So why they are primed? Because they also come from the inner cell mass. Well, actually we investigated a few years ago. When you isolate the ICM and 2 weeks afterwards you have your stem cell colonies, we investigated what happens in the meantime. The interval between the inner cell mass and your eventual stem cell colony. So we investigated these cells that arise. We call this the PICMI stage, which was published in 2 journals. So we saw that between ICM and stem cell colony there is a phase that we named the PICMI, so the post inner cell mass intermediate. And when we molecularly analyzed these PICMIs, we showed that they contain a lot of late epiblast markers. So actually the inner cell mass undergoes first some differentiation towards late epiblast, and then finally the stem cells that you have, actually they are similar to the mouse epiblast stem cells, so they are primed. But because the success in mouse, for example, to make gametes, was only possible with the naive ones. They also tried it with the epiblast stem cells, and it was not possible to get functional sperm or oocytes, so only possible with the naive ones to make gametes. So the race was open to also make in human naive human embryonic stem cells. And now already 10 to 15 groups have recently published protocols to make naive embryonic stem cells, so we can also make them in human. Jakob Hanna was the first one already in 2010, his group, and then yeah, all other big stem cell groups also succeeded in making naive embryonic stem cells. This table shows just all small molecules and growth factors that they add to the medium to have these naive embryonic stem cells. Well, mostly they convert primed stem cells, they put them in these specific culture conditions, and after a while you have the naive ones. Only 3 groups succeeded directly from the blastocyst stage to produce these naive embryonic stem cells. But the efficiency is very low. Mostly they don't report how many blastocysts they need to make one embryonic stem cell line, but one report did, and they needed 158 blastocysts, good quality blastocysts, to have one stem cell line. So the process is still very inefficient for the moment. And now, yeah, in the big stem cell groups, yeah, they show their testosterone and they claim my embryonic stem cells are the best, mine are the best. So now this discussion is going on. They do a lot of this molecular analysis to show that they have the best naive embryonic stem cells. We also have our naive embryonic stem cell cocktail, which was in 2015. So also morphologically you can distinguish them. The primed ones are flat colonies, the naive ones are domed colonies. So we think that our stem cell cocktail is of course the best one, which we are now proving. So the last step in human, is this possible to make? embryonic stem cells towards gametes. Is it possible? This is nice. If you're interested, I can recommend this review. It shows a nice overview of what recently have been done to make stem cell derived gametes, not artificial gametes from stem cells, from the group of Carlos Simon. Well, it's a very, a very complete overview of all the studies that have been done. So they succeed from embryonic stem cells to make primordial germ cells. That's basically it. So they prove with all markers, gene expression markers, that the cells that they have after some differentiation show the characteristics of primordial germ cells. We also showed primordial germ cell formation from embryonic stem cells, and actually we showed also if you derive embryonic stem cells in a certain medium with activin A, it already promotes your eventual differentiation towards primordial germ cells. So yeah, we also have some VASA-positive cells in our cultures, which is a characteristic of primordial germ cell formation. Again, the group of Jacob Hanna from Israel, and in collaboration with the group of Azim Surani, which is one of the pioneers also in the stem cell field from Cambridge. They have, last year, published a study in which they started for the first time from naive human embryonic stem cells. So all the previous studies were done with primed human embryonic stem cells, but they were the first to try it also in naive human embryonic stem cells. And they showed that they could very efficiently get to the first step, so to the primordial germ cells. They also showed that some differences with the mouse, or for example SOC17, which is not involved in mouse PGC formation, is one of the key regulators in human. So it means that we cannot translate all the experience we have in mouse to the human, which is of course makes it a little bit difficult. I also welcome you tomorrow. He's on the main stage, Jacob Hanna, so the senior researcher from this group. He will talk about also the naive pluripotent stem cells, if they are the key to success to in vitro gametogenesis. Well, just a slide to show that people already use cells derived from human embryonic stem cells, not for reproduction, of course, because we don't have the gametes yet. But for example, for this macula degeneration, so it's kind of eye disease, Stargardt syndrome, they already use retinal cells derived from embryonic stem cells, and they transplant them back already to patients. So already clinical studies happen with cells derived from embryonic stem cells. I think it's a little bit premature to do this, to be honest, because we just discovered this difference between primed and naive, so they work of course with the primed ones because the naive ones were not existing at that time. So I think it's too premature yet to do any clinical trials with cells derived from embryonic stem cells, because we know too less about them still. So I would recommend some caution to use them already. So will the naive be better for clinical applications? Well, there are some indications from animal models that they are. If you compare embryonic stems, naive ones versus primed ones, that they are more superior in differentiation potential. Also in the iPS cells, if you make them naive, you have better differentiation possibilities. The ladies are there. Do I still have some minutes or 2 minutes? Okay. So the most recent breakthroughs in stem cell derived gametes. So if you remember from the Hayashi studies, they still needed this in vivo step, but now in February this year, they succeeded to do it fully in vitro. So no in vivo step anymore required. They could get functional sperm just in the petri dish. So from embryonic stem cells, the whole way to spermatozoa, they could do it fully in vitro. It was a Chinese group, February 2016. These were the media headlines. So the key was that they co-cultured them with postnatal testicular cells. So They did a co-culture, and of course they added some factors which were important for germ cell formation. They didn't get functional, well, they got functional sperm, but the sperm, it was just the stage before, so the spermatid stage, they didn't have a tail, so it's the round cells. But they injected this into oocytes and they could get offspring also using these spermatids. Efficiency from offspring was a little bit lower compared to control, 3 versus 10%, but yeah, their control was a little bit low. Normally you get a little bit higher offspring if you do hESC and you do embryo transfer. So yeah, the spermatids gave a little bit lower offspring, but they could get offspring. I will skip this paper from Carlos Simon. So, conclusions, take-home messages. Which technique will be better? So, the cloning or the iPS cells? Should we start from the naive ones, or maybe the primed ones will still be better? So, this is still under investigation. And the naive ones, yeah, you have plenty of naive conditions. Which one should we use? Of course, the safety. We should be very careful if you use this clinically, that we are very aware from the safety. So for example, the first study that got sperm, all the mice died within 5 months. So they saw some epigenetic abnormalities in the mice, in the offspring. That's the last slide. So I would really strongly advise that it will be possible. People always ask the time period. Let's say 10 years, it will be possible in human. So I would strongly recommend, before we clinically apply it, to do functional testing so that we make embryos, that we check if they can induce activation, fertilization, embryo development. Nowadays, molecular testing, it's evolved so much, we can do it really on the single cell stage, so we can check genomics, epigenomics, gene expression on single cells, on the gametes, or on the produced embryos, so we should do a lot of these tests. And actually, only in the mouse it has been succeeded, but I also would strongly advise that we also try to invest more in other animal models, that we see the safety issues over there. Okay, ethical reflections, I will not touch them because of time limit, but of course, it's ethically very controversial also to make stem cell gametes, so this should also be tackled by the ethical groups, and for the future we need to fully do it in vitro in human. We need to optimize the culture conditions, safety testing, so a lot of omics we should do, and some ethical reflections. Finally, we have a workshop on one of these topics is stem cell derived gametes, so I welcome you all in Amsterdam. It also will be about mitochondrial transfer, also about germ cell, the genomic editing in germ cells, so I welcome you all very much to Amsterdam in September, and this is the team I'm working in. So thank you for your attention.
Thank you. So this talk is now open for questions from the audience.
You can wake up again.
So I would like to start with a question. So it seems to me that you made it quite clear that we're not really ready yet to use stem cell to treat infertility, at least gametes. However, there are some treatment with stem cell in the ovary that have been used, that are being used in several countries. Some of them within the context of a supposed clinical trial, and some of them just proposed to patients. So what's the deal with that?
Well, they are of course they are also doing research. So instead of starting from the embryonic stem cells, they start from the spermatogonial stem cells or the oogonial stem cells, which there is still some controversy if they exist or not. I'm not aware that already someone succeeded fully in making functional sperm cells from spermatogonia still. They are testing it. The group of Brussels is testing this option, but as far as I know, they are not yet to the stage that they have functional sperm which they have used for clinical trials yet.
No, I was referring to improvement of oocyte quality.
Sorry?
I was referring to the improvement of oocyte quality.
Oocyte quality?
Yeah.
And how do you mean, Rita?
With these supposed ovarian stem cells that are manipulated and then used to...
I know what you refer to. So, but in this case, they harvest mitochondria from this oogonial stem cells, from the oogonia, and this mitochondria they inject into mature oocytes to rejuvenate them, so to give them some fresh mitochondria. They have done this in a clinical setting, so in some papers they have shown that it would improve RT outcomes, so it would improve implantation rates and so on. But the studies that I read were not very convincing to me. So the control groups were not very well matched. So I don't think you can attribute at this point their success to the fact that they used this mitochondria to rejuvenate the oocytes. Also in animal models there is no evidence that really have shown that if you harvest mitochondria from oogonial stem cells or whatever, that you can improve implantation rate or that you can improve oocyte quality. So also in animal models there is no evidence at all that this would improve ART outcome. So for me these studies are also a little bit controversial and also a little bit too premature to already clinically apply, if I must be honest.
Yeah, thanks.
Bjorn, I also have a question on the way that you, in the stem cell research, you all have your own soup to make the cells. So how do you relate that compared to the whole issue that we had on culture media and birth weight of children and things like that? And if you look at all the different kinds of compounds that you have to put into the systems to get these cells, what is your opinion on the way that the stem cell society is going to go about, introducing this in a clinical setting, since there's already been such a huge issue about the culture media in IVF?
It's a good point, Kelly. Well, to get our naive embryonic stem cells, we have to add a lot of small molecules which target all molecular pathways. And for some of them we know exactly which pathway they target, but you have downstream also a lot of downstream processes that we cannot control or we don't have full insight yet in what's happening. So it's a relevant question. Well, the stem cells that we finally have, we always compare them to in vivo counterparts, so like to the ICM. So if we prove on the gene expression level and also on the epigenetic level that they are comparable to in vivo counterparts, I think, we can say they are more or less safe. Of course, the embryonic stem cells, we will not put them back, especially for reproduction. We don't have to put back also the differentiated cells. We don't have to put them back in the patients. We can do the fertilization in the dish. So that's one advantage for reproductive purposes, that you don't have to put back the differentiated cells. But I would strongly advise, to do a lot of molecular testing on the stem cells itself, but especially on the end product, the final gametes, the sperm or the oocytes. We have a lot of in vivo produced sperm or oocytes, so we can do a very in-depth comparison between the in vivo produced end product sperm or the oocyte and the in vitro generated sperm or oocytes.
Okay, so there's no other question? Thank you very much, Bjorn.
Okay, thank you.